Today, let us have a discussion on antimicrobial agents. In antimicrobial agents, let us discuss the first topic, that is cell wall synthesis inhibitors. Cell wall synthesis inhibitors. Now, this is the first category of antibiotics, that are cell wall synthesis inhibitors. Before discussing about the cell wall synthesis inhibitors, a student should have the clear idea of the composition of cell wall of bacteria or any microbes. So now let us discuss the composition of cell wall of any bacteria. That may be either gram positive bacteria or else gram negative bacteria. There will be slight difference in the thickness of the layer of cell wall in case of gram positive and gram negative. In case of gram negative bacteria, there will be less thickness of cell wall when compared to that of gram positive bacteria. Now let us have a discussion of, let us have a microbiological part of cell wall. Before going into detail of cell wall, we need to have a clear idea of what is an antibiotic. Antibiotic is a drug which is derived from the microorganisms and used to treat against the same microorganisms. This is the definition of antibiotic. It is a drug which is derived from the microorganism and used to kill or inhibit the same microorganism is called antibiotic. And a clever student should remind that antibiotics not only used to treat the microbes, they are also used to treat some other diseases like cancer. In that cancer, antibiotics are also used. So that is about antibiotic. Now we will have a discussion on cell wall, the composition of cell wall, how the cell wall is made in case of bacteria. Now let us consider before going into detail, let us consider this is a cell membrane of gram positive bacteria. Outside that cell membrane, we have cell wall. This is a cell wall and this is a cell membrane. Now, what is the actual mechanism taking place in the cell wall synthesis inhibitor means the outermost layer is cell wall. Inner to the cell wall, we have cell membrane. Now, these cell wall synthesis inhibitors are inhibiting the cell wall synthesis. Like, there will be inhibition of cell wall synthesis. If there is inhibition of cell wall synthesis, the remaining part is only cell membrane. So, when only the cell membrane is remained, there will be influx of solutes like sodium as well as water. There will be influx of sodium as well as water, and there is rapid influx of this sodium and water will result in the increase of size of this bacteria, and there will be lysis of bacteria. Ultimately there will be lysis of this bacteria. This is the mechanism which is taking place behind this cell wall synthesis inhibitors. Now let us have a discussion on the composition of cell wall. Either it may be gram positive bacteria or gram negative bacteria. Let us consider this is a cell membrane. Let us consider this is a cell membrane. Now, there are some structural and functional units of cell wall synthesis. There are some structural and functional units of cell wall. They are made up of, they are called as peptidoglycans. Peptidoglycans are the functional units of the cell wall of bacteria. Now, in other words, we can say that this cell wall of bacteria is made up of two components. One is peptide bonds and other one is glycans. Glycans are nothing but sugar molecules. They are made up of two components. One is sugar molecules along with peptide bonds. Now, first let us discuss about these two parts. One is peptides and other one is glycans. First let us discuss about what are glycans means. We have two units like N-acetylglucosamine. Now these N-acetylglucosamine should convert into N-acetylmuronic acid. N-acetylglucosamine should be converted into N-acetylmuronic acid. So NAD and NAM, these are two units of cell wall of bacteria. Now this NAD should be converted into NAM. Now there is an enzyme called, there is some substrates or enzymes which are responsible for the conversion of N-acetylglucosamine into N-acetylmuronic acid. That substrate is called phosphoenol pyruvate. That is called phosphoenol pyruvate, or else enol, another enzyme called enol pyruvate. Phosphoenol pyruvate or enol pyruvate. Now this is the first step which is taking place, conversion of N-acetylglucosamine into N-acetylmuronic acid. After this, there should be attachment of N-acetylmuronic acid with that of N-acetylglucosamine. Now here N-acetylmuronic acid should be attached with N-acetylglucosamine. This is the glycan part of this cell wall. We have completed the glycan sugar. We have completed the synthesis of these sugar molecules. Now coming to the peptides. Peptides are nothing but proteins. How these proteins are made means, there is an enzyme called, it's an amino acid called L-alanine. Now this L-alanine should be converted into D-alanine. L-alanine, levoisomer of alanine, should be converted dextroisomer of alanine. So there is an enzyme which is acting at this site for the conversion of L-alanine into D-alanine. So what is the enzyme which is responsible for the conversion of L-alanine into D-alanine is alanyl racemase. Alanyl racemase is the enzyme which is responsible for the conversion of L-alanine into D-alanine. Now let us consider there are some D-alanine molecules here. Let us consider 5 amino acids of D-alanine are present here. Now these amino acids, D-alanine amino acids, should be attached with each other with some kind of a bond. So there is a peptide bond. Before going into that, okay, there is a peptide bond which is connecting these amino acids with that of NAG units. These amino acids are connected to NAG units with special bond. Now there is an enzyme which is responsible for the attachment of these peptide bonds with that of NAG units. That enzyme is called alanyl ligase. Alanyl ligase is an enzyme which is responsible for the attachment of peptide bonds with that of NAG units, and the another function of this alanyl ligase is that helps in the attachment of glycine with that of D-alanine. So this is the glycine and this is the D-glycine and this is the L-alanine, D-alanine. So in this way there is a synthesis of protein will be taking place. So this is the complete one unit of cell wall. Now this entire process is taking place within the cell membrane, and it should be thrown out of the cell membrane and it should come out of the cell membrane. How this unit of cell wall will be coming out of this cell membrane means, there is a transporter called, let us consider, this is a transporter which is present on cell membrane, on outer side of cell membrane. The name of this transporter is lipid transporter, but scientifically the name of this is bactoprenol. Shortly called as bactoprenol pyrophosphate, BPP. The name of this is bactoprenol. Now, the function of this bactoprenol is it helps in the translocation of these units of cell wall from inside of this cell membrane to the outside of cell membrane. Now, let us consider these are came out with the help of this bactoprenol, and this is the NAG unit, and this is the NAM unit, and this is one unit, and NAG attached to NAM unit. This is another. These are two units of cell wall. Now there is a attachment of, there is a peptide bond which is present between these two. Like one bond is formed between this D-alanine with that of another D-alanine. So there is an enzyme called which is responsible for the formation of peptide bond. The name of that enzyme is transpeptidase. Transpeptidase is the enzyme which is responsible for the formation of peptide bonds. Now, this is the structure of cell wall. In this way, there will be complete formation of cell wall will be taking place. This is the synthesis of cell wall. First of all, there will be synthesis of glycan molecules, and there will be synthesis of sugar molecules, and they will be covered with the help of bactoprenol pyrophosphate. And from this, when it reaches out, there will be attachment of peptide bonds between these two units, and simultaneously these these bond formation will be taking place throughout all the cell wall. The enzyme which is responsible here is transpeptidase. Now, this is all the microbiological part of cell wall. Now, coming to the pharmacology which is involved here is, now how we can inhibit the cell wall synthesis means, let us consider the first possibility how we can inhibit the cell wall synthesis means. The first one is we can give the analogs like phosphoenol pyruvate. This is the first category. Phosphoenol pyruvate analogs. Analogs of phosphoenol pyruvate. So what are the drugs which comes under this PEP analogs means? First drug is phosphomycin. Second drug is phosphidomycin. Phosphomycin and phosphidomycin. These are the two drugs which comes under the phosphoenol pyruvate. What will be the mechanism means? These are the analogs of phosphoenol pyruvate, and there will be competitive inhibition between this phosphomycin and phosphidomycin and phosphoenol pyruvate. In the case of this phosphoenol pyruvate, these phosphomycin and phosphidomycin will be coming and attached to the substrate, and there will be competitive inhibition of this phosphoenol pyruvate, and there will be no conversion of NAG unit into NAM unit, and there is no formation of this cell wall takes place. This is the first, let us consider this is the first method to inhibit the cell wall synthesis. After this inhibition of cell wall synthesis, first one, what is the second type means? We are completed the phosphoenol pyruvate. What is the second most possibility here is we can inhibit this D-alanine molecule. We can inhibit the D-alanine molecule second possibility. We can inhibit the D-alanine molecules. If there is inhibition of D-alanine molecules, they cannot form this protein. Ultimately, there will be no attachment of glycine molecules, there will be no formation of protein molecules. So, what is the drug which comes under the inhibitors of D-alanine? I will call cycloserine. Cycloserine is a drug which comes under the inhibition of D-alanine molecules. This is a second category. Now, we have completed this first one and second one. What is the third most possibility is, we can inhibit this action of this bactoprenol. We can inhibit the action of this bactoprenol. This is a third possibility which helps in inhibiting the synthesis of cell wall. If this bactoprenol pyrophosphate is inhibited, whatever the NAM units and AG units, whatever the unit of cell wall which is formed, cannot translocate from inside the cell membrane to the outside of cell wall. Inside of the cell membrane to the outside of the cell membrane. As a result, there will be no formation of cell wall. So, third category is bactoprenol pyrophosphate inhibitors like BPP inhibitors. What are the drugs? What is the drug which comes under inhibition of BPP? Means bacitracin. B for bacitracin. Penicillins is a drug which inhibits the action of this BPP. Now we have completed that BPP, and coming to the next category means so these whatever the drugs cycloserine which is given, it is the D-alanine analog. It is not an inhibitor. It is an analog of D-alanine. Like here. It is an analog of D-alanine, and there is competitive inhibition between this cycloserine and D analog. Same mechanism which is taking place in case of phospholipid pyruvate will be taking place here. This is the second one, and this is the third one. Coming to the fourth category, like D-alanine inhibitors. This is the fourth category. Like we will inhibit the D-alanine molecule. If it is inhibited, there will be no formation of these protein molecules. So what are the drugs which comes under D-alanine inhibitors? Like the drugs which comes under these D-alanine inhibitors are glycopeptides. So the drug which comes under is vancomycin. Vancomycin is the D-alanine inhibitor. This is the fourth category. And coming to the last category, that is, we have completed all this. The remaining part is only transpeptidase. We can inhibit this transpeptidase. This is the fifth category. We can inhibit the transpeptidase. If there is inhibition of this transpeptidase, there is no formation of peptide bonds between the units of cell walls. So, what are the drugs which comes under this transpeptidase inhibitors? Are like first drug is beta-lactams. Beta-lactam antibiotics. Next one is beta-lactams are nothing but, beta-lactam antibiotics are nothing but, these are the penicillins. It includes penicillins. It includes cephalosporins. And it includes monobactams. It includes monobactams, and last category it includes carbapenems. So this is the category of cell wall synthesis inhibitors. We have discussed the inhibitors of cell wall only in a normal way. We have not gone into depth of this any of the drugs. So this is a clear picture of inhibition of cell wall synthesis. Now we will go into the detail of, first we will go into the detail of penicillins. we will discuss about the penicillins. So penicillins are the first antibiotics to be discovered. So now let us discuss the classification of penicillins based on their spectrum. Based on their spectrum, penicillins are classified into two categories. One is narrow spectrum penicillins, while the other one is broad spectrum. These are the two spectrums of antibiotics penicillins. One is narrow spectrum penicillins and the next one is broad spectrum penicillins. So what is the difference between these two spectrums? We will come to know after some time. First let us discuss the first drug which comes under the narrow spectrum antibiotics, like penicillin G. Penicillin G is the first narrow spectrum antibiotic, and as we know that it is limited for only gram positive bacteria. It cannot show any activity on gram negative bacteria. Why there is no action on gram negative bacteria and why it is only limited to gram positive bacteria means we will have a discussion on it. Let us consider this is the structure of gram negative bacteria and this is the cell wall, and inside we have cell membrane. Now on surface of cell wall we have like some porins. These are called as porins. On surface of cell wall we have porin like structures. Whenever any drug, any antibiotic will be given to this bacteria, so when it is showing the action, it must have two function, like two characteristics. The first characteristic of an antibiotic should it must be small size. It must be of small size. And next characteristic is it should be water soluble. These are the two important characteristic features of any antibiotic which are acting on gram negative bacteria. So whenever these two are whenever the drug satisfies these two categories, these two characteristics, it will show its action on enzyme called transpeptidase. So what is the drawback here is this penicillin is lipid soluble. Here it is a water soluble, but this penicillin G is lipid soluble. It is a lipid soluble and it is limited to only gram positive bacteria. Now, how we have modified the narrow spectrum antibiotics like penicillins into the broad spectrum antibiotics means. So, let us have that discussion here. Now, as we all know that this is the structure of penicillin, and we have an R group. This is a beta-lactam ring and this is a thiazoline ring. So this is a beta-lactam ring and this is a thiazoline ring. Beta-lactam ring and thiazoline ring and one R group. So what happened? What is the changes which is taking place while moving this narrow spectrum to the broad spectrum means, so how this narrow spectrum is converted into broad spectrum means, there is a change in the R group. There will be alterations in the R group. We will remove that R group, and whenever we remove that R group, in place of R group we will substitute the another groups. So this is the difference between the narrow spectrum penicillins and broad spectrum penicillins. In case of broad spectrum penicillins, we are removing the R group. In place of R group, we are adding another functional group. So what are that another functional group means? We have 3 functional groups. Like in case of broad spectrum antibiotics, we are having 3 functional groups are adding in case of this R group. So what are the 3 categories, 3 functional group means? First one is, in case first category is amino group. The first category is, in case of R group, in place of R, amino group will be substituted. This is the first category. And second category is, in place of R group, we will substitute carboxyl group. This is the second category. And third category is urido group. Urido group. So these are the 3 functional groups which are substituted in the place of this R group. One is amino group, next one is carboxyl group, third one is urido group. Now these amino, they have some charges on it. These amino group have positive charge, whereas these carboxyl group have negative charge, whereas this urido group have both positive as well as negative charge on it. Positive charge, negative charge. So if we think logically, out of these 3 groups, this urido group of penicillins are better showing action on gram negative bacteria when compared to amino group and carboxyl group. So what are the drugs which are included under amino groups are like amoxicillin, and ampicillin. Amoxicillin and ampicillin. So amoxicillin and ampicillin, these are 2 drugs which are included under these amino groups. So these amino groups are orally used wide spectrum antibiotics. So these are orally used wide spectrum antibiotics, and these are the drug of choice for listeria and hemophilia influenza, like pneumonia, and next one is enterococci. And these are the orally used drugs. Coming to the carboxyl groups, so what are the drugs which are included under here is carbacillin and ticarcillin. Carbacillin and ticarcillin, these are the 2 drugs which are included under this carboxyl group. So these are used parenterally, while these are used in oral manner, these are used in parenteral manner. And what are the drugs which are included in this urido group are piperacillin, azlocillin, and meslocillin. Piperacillin, azlocillin, and meslocillin. Now these 2 groups are taken in the form of parenteral routes. These 2 group of drugs are taken in the form of parenteral routes, like carboxyl group antibiotics and urido group antibiotics are taken in the form of parenteral routes. So these are used to treat like these two drugs, piperacillin and ticagrelor. These two drugs are used to treat pseudomonas. So these urido group of drugs and carboxyl cyclic group of drugs are called anti-pseudomonal drugs. Anti-pseudomonal antibiotics or else anti-pseudomonal penicillins. Anti-pseudomonal penicillins. Now, this is about some classification of broad spectrum antibiotics. Coming back to the penicillin G, it is, if I discuss some pharmacokinetic action of this penicillin G, it is less or it is poorly absorbed. There will be poor absorption if we take in oral manner. Now, what is the reason behind this poor absorption of by oral manner means there is a pumps called P-glycoprotein pumps, and it is a short acting because of there is a P-glycoprotein pumps present in the nephron. It is results in the secretion of this penicillin G, and there will be excretion of this penicillin G, and these are oral there is poor absorption if we take these drugs in oral manner. Why? Because they are damaged or they are digested with the help of gastric juices. So in order to overcome this digestion with that of gastric juices, we have made another drug like penicillin V. This is penicillin G and this is penicillin V. This is acid stable antibiotic. This is acid stable penicillin, and as I told that these are parenteral route of administration, and this is short acting antibiotics, and in order to increase the action of this penicillin G or penicillin V, we have to add a salt like first one is benzathine penicillins. So this benzathine is a water soluble drug, and this is a drug of choice for rheumatoid fever. And it will show its action for 28 days. We have is 28 days, and this benzathine is an antibiotic which is used in case of for the treatment of rheumatoid fever, where the patient will take this benzathine once in a month. This is 28 days. Now, this is about penicillin G and penicillin V. If we come to the uses of this penicillin G and penicillin V, it is used to treat against the gram-positive bacteria, and it is limited to only gram-positive bacteria. And there is an exception like meningococcus. There is an exception for meningococcus, which is a gram-negative bacteria. Like these penicillin G and penicillin V can show effect on meningococcus bacteria, and it can show its action on syphilis and streptococcus and leptospirosis. So this is some information about classification of penicillins like narrow spectrum penicillins and broad spectrum penicillins. In broad spectrum penicillin, this R group is substituted by another amino group, carboxyl group and urido group. Now let us discuss some mechanism of resistance. let us discuss the resistance for the penicillins. The resistance for the penicillins are studied under two categories by two ways. One is the resistance developed in gram-negative bacteria. In case of gram-negative bacteria, and next one is in case of both gram-positive and gram-negative bacteria. These are two categories or two classifications by which we can study the resistance of penicillin. One is by gram-negative bacteria and one is gram-positive bacteria. First, let us discuss how the resistance will be developed only in case of gram-negative bacteria. Now let us consider cell membrane, and this is a cell wall, and there is a porins, as we know that these are the porins, and on surface of this cell membrane we have this is a transpeptidase, these are the porins. So whenever the antibiotic will be given, that antibiotic will enter through the porins, they will enter through the porins and will reach this transpeptidase enzyme, and resulting in the inhibition of this transpeptidase enzyme and result in the inhibition of protein synthesis. So this is the normal mechanism which is taking place in case of gram-negative bacteria. How the resistance will be developed in case of gram-negative bacteria means the first type of resistance is there will be decrease in the number of these porins. That means there will be decrease in the number of porins. If the porin number is decreased, whatever the antibiotic which is given, they cannot enter into the into this middle area, periplasmid area. So whenever it is not entered into this area, they cannot attach to this transpeptidase enzyme. If it is not attached to transpeptidase enzyme, there is no protein synthesis inhibition. In this way, the number of porins will be decreased. This is the first mechanism of resistance. So this decrease in number of porins can be seen in special microbes, special category like Pseudomonas. This is the first type of resistance which is developed within the gram-negative bacteria. That there will be decrease in the number of porin productions. And next one is whenever the drug will be entered into this area, there are some efflux pumps are present on surface. On surface of, let us consider, on surface of this cell wall, there are some efflux pumps are present. Whenever this drug is coming inside this periplastic area, it will reach the efflux pump. With the help of efflux pump, whatever the drug which is given will be effluxed out. In this way, there will be efflux of this drug, and there will be no attachment of this drug with that of transpeptidase enzyme. Ultimately, there is no inhibition of protein synthesis. This is a second type of mechanism like efflux pumps. Efflux pump. So this type of mechanism of resistance can be seen in case of gonorrhea and Pseudomonas. Gonorrhea and Pseudomonas. So this is the two mechanisms which is taking place especially in case of gram-negative bacteria, which results in the resistance developed in case of gram-negative bacteria. Coming to the gram-positive and gram-negative bacteria, common for both gram-positive and gram-negative bacteria. That is the first. What is the first step means? We have discussed about porins and we have discussed about efflux pumps. The next possible site is transpeptidase enzyme. There will be alterations in the transpeptidase enzyme. there is alteration in the transpeptidase enzyme. So if there is alteration in the transpeptidase enzyme, this whatever the drug which is entered cannot attach to this transpeptidase enzyme. So there will be non-cyclic inhibition, non-competitive inhibition will be taking place. So this can be seen in case of Staphylococcus. It can be seen in case of Staphylococcus aureus. It results in MRSA. So this is the first category, and coming to the next, this is the first, second, and this is the third mechanism of resistance. And coming to the last and most important type of resistance is there will be synthesis of beta lactamase. Synthesis of beta lactamase. So what is beta lactamase? Beta lactamase is an enzyme, or it is an enzyme which is synthesized by the bacteria, which will inhibit the which will kill the beta lactam ring of an antibiotic. Let us consider this is the structure of penicillin, and at which side this beta lactamase enzyme will be acting means it will be acting at the site of this base. They will act at the site of the base and result in the cutting of the base. So there is a classification of beta lactam synthase, beta lactamase synthesis. What is that classification means? There are 4 types of beta lactamases are present. So this should be clear for everyone. There are 4 classifications, or there are 4 different types of beta lactamases are present, which are developed by the bacteria. What are the 4 means? First one is expanded spectrum beta lactamase, ESBL, extended spectrum beta lactamase. Why it is called extended spectrum beta lactamase? This is the first category, right? Extended spectrum beta lactamase. What is the speciality of this extended spectrum beta lactamase means? They will produce the beta lactamase, like first one produces against the penicillin. The effective enzyme is penicillinase. They will synthesize the penicillinase and resulting in the resistance in the penicillin, and they will result in the stimulation of, they will result in the synthesis of cephalosporinase, and resulting in the resistance developed in the cephalosporins, and they will also result in the synthesis of monobactamase. So these extended spectrum beta lactamase will show effect on penicillins, cephalosporins, and monobactams. So my question is, as I told, these extended spectrum beta lactamases are showing the action on penicillins, cephalosporins, monobactams by producing penicillinases, cephalosporinases, and monobactamases respectively. So for this extended spectrum beta lactamase, what is the drug of choice? The answer will be lying in this only. It will show action on penicillinases, it will show action on cephalosporinases, it will show action by monobactamases. So the drug of choice for these extended spectrum beta lactamases is, drug of choice is, as we discussed in beta lactamases, it includes 4 categories. One is penicillins, cephalosporins, monobactams, and last one is carbapenems. So whenever person suffering from extended spectrum beta lactamases, so the drug of choice for extended spectrum beta lactamases is, the remaining one is carbapenems. This is an important multiple choice question to be remembered by a clever student. For ESBL, the drug of choice is carbapenems. This is the first category of beta lactamase. So coming to the second category, it contains all the enzymes like penicillinases, cephalosporinases, carbapenems except monobactamases. Except they contains all the, they will show resistance against penicillinases, cephalosporinases, carbapenems, but they cannot show the resistance against this monobactamases. They cannot produce this monobactamase and cannot show the resistance against monobactam. This is the second category. Third category is they have only one type of enzyme and they will show resistance to only one type of antibiotics like cephalosporins by producing cephalosporinase. Listen carefully. Extended spectrum beta lactamases will show effect on these three organisms, and the second one is second group of beta lactamases will show effect on penicillin, cephalosporin, and carbapenems. They cannot show effect on beta lactams. And third one cannot show effect on. They can show effect only on cephalosporins by producing cephalosporinase. And next one is the last fourth category of beta lactamases is cloxacillinase. Cloxacillinase, they will inhibit the, they will show the resistance against cloxacillin. So this is the action, this is the reversible action which is shown by the bacteria by producing the beta lactamases. Now many scientists thought that how to overcome these beta lactamases, and they made researches and they have studied and they came with a new ideas. Like we will study that what are new ideas means. So as we know that while we giving the antibiotics, these bacteria are producing the beta lactams and inhibiting the beta lactams, action of beta lactamase by inhibiting the beta lactams. So scientists had made a research and came with a new type of drugs. So that will be the action of human being on bacteria. So that is a reverse action of human being on bacteria. Like because as this bacteria is synthesizing the beta lactamases, the first category is what is the reverse action of doctors on beta lactamases is, the first category is they have discussed beta lactamase inhibitors. Beta lactamase inhibitors. So bacteria is showing or it is stimulating the beta lactamases. Counter to that, we are giving the beta lactamases inhibitors. So what are the beta lactamases inhibitors means? First one is clavulanic acid. So there will be always a combination of these beta lactamases with antibiotics like broad spectrum penicillins. Now this clavulanic acid will always combine with amoxicillin. They will be always combined with amoxicillin. This is the first combination. Coming to the second combination is this is clavulanic acid. Next one is sulbactam. This sulbactam will be combined with ampicillin. They will be combined with ampicillin. And third category of beta lactamases inhibitors are tazobactam, now this tazobactam will be combined with piperacillin. They are combined with piperacillin. So this is the first category. that is our counter reaction for the beta lactamase synthesis. We are inhibiting the beta lactamase enzyme. This is the first category, and next category is by producing the penicillinase. So these drugs are also called penicillinase. So what is the mechanism will be taking place here means there will be change in the R group. Like if we let us consider this is the structure of penicillin, and when we give the penicillinases, there will be alterations in the R group. There will be alterations in the R group, and there will be structural changes in the penicillin. Now these are called penicillinase resistance, penicillinase resistant penicillins, penicillinase resistance penicillins. So penicillinase resistance penicillins. So these are also called anti-staphylococcal antibiotics. Or else anti-staphylococcal penicillins. What are the drugs which comes under these penicillinases or penicillinase resistance penicillin? There are oral drugs and there are parenteral drugs. What are the oral drugs like oxacillin and cloxacillin, and next one is dicloxacillin. Oxacillin, cloxacillin and dicloxacillin. So what are the drugs which comes under parenterals are methicillin, and neficillin. Methicillin and neficillin, these are two drugs which comes under parenteral penicillinase resistance penicillins. So there are some side effects important side effects of this oxacillin is the results in hepatotoxicity, and the side effect of dicloxacillin is it is the most potent penicillinase resistance penicillin. And next one is coming to the side effect of neficillin is it results in neutropenia, and coming to the side effect of methicillin is it results in interstitial nephritis. So these are some counter actions by doctors on bacteria. So this is the action by doctors on bacteria. So bacteria is also clever one. Again, the bacteria is showing its reverse action on these drugs. So what is the reverse action on these drugs means we will discuss about that. When we give beta lactamase inhibitors and penicillinase resistance penicillins like oxacillin, dicloxacillin, cloxacillin, and methicillin, neficillin. So what is the next step which is taken by bacteria means that bacteria is also clever one. Now what is the reversible step which is taken by the bacteria on these drugs means there will be there is some alterations which are taking place in the transpeptidase. Transpeptidase is also called penicillin binding protein. This is the reversible action on, that is counter action of bacteria on this beta lactamases and penicillinase resistance penicillins. So when we once again there is counter action of bacteria for beta lactamase inhibitors and penicillinase resistance penicillins. So what is the counter action of these bacteria on beta lactamases inhibitors and penicillinase resistance penicillin is there will be alteration. This bacteria will be making alterations in the penicillin binding proteins. Alterations in penicillin binding proteins. This is the counter action of bacteria on beta lactamase inhibitors and penicillinase resistance penicillin. So how can we treat? What is the next result in the alterations of protein penicillin binding protein means? So if there is, let us consider it's a graminate cell membrane and cell wall. On the surface of the cell membrane we have transpeptidase. So there is alterations in this transpeptidase enzyme. So if there is alteration in the transpeptidase enzyme, these antibiotic like penicillin cannot enter and they cannot show its action on transpeptidase enzyme because there is alteration in the protein binding protein, penicillin binding protein. So then these results in the, let us consider, it results in the MRSA. MRSA. So what it results into the MRSA. Now what is the action, sorry, what is the treatment of drug of choice for the MRSA means we cannot do anything with it of transpeptidase. So next our option is simply going into the another like alternate methods to inhibit the cell wall synthesis. We cannot do anything with that of transpeptidase. Now we can inhibit, next options are we can inhibit phosphonyl pyruvate, or we cannot go to the transpeptidase, we can inhibit D-alanine and okay. Now for MRSA we cannot go or we cannot do anything with that of transpeptidase because there is alteration in the transpeptidase enzyme. The next only option for the treatment of MRSA is we can inhibit D-alanine. We can inhibit only D-alanine molecule in case of MRSA. So what is the drug of choice for MRSA which is inhibiting the D-alanine means the drug of choice is, what is the drug which inhibits the D-alanine is vancomycin. So this alteration in the penicillin binding protein is the counter action of bacteria. So our doctors are much intelligent than this bacteria and they have worked again and they studied about this MRSA and they gave the drug of choice as vancomycin. So this is the counter action of doctors. So because of counter action of this these doctors, there is resistance developed for this vancomycin also. So now this vancomycin is inhibiting this D-alanine molecule. Now this bacteria is more dangerous than doctors. So these doctors gave the drug of choice for MRSA as vancomycin. Then the next counter action of bacteria for this vancomycin is this bacteria result in the alteration in the D-alanine molecules. Why? Because this vancomycin is showing the action on D-alanine. Then there is alteration, this is the counter action of bacteria on vancomycin, that is alteration in D-alanine. There is alteration in the D-alanine molecule. Now this is the alteration in the D-alanine molecule, then this vancomycin cannot show its action. Here the transpeptidase like enzyme got altered and D-alanine got altered. So we cannot do anything without of cell wall. Now, what is the? It results in a condition called VRSA, that is vancomycin resistance Staph aureus. This is methicillin resistant Staph aureus. So in VRSA there is alteration of D-alanine molecule. We cannot do anything with the D-alanine and cell wall. This transcriptase. We cannot do anything with that of cell wall. Now, what is the another option for this vancomycin resistant Staph aureus? The only option is we cannot do anything with that of cell wall. The only option is we can do something with that of only cell wall. So we can inhibit the cell wall. How we can inhibit the cell wall in case of vancomycin? So what is the drug of choice for VRSA means? Drug of choice is cell membrane inhibitors. Cell membrane inhibitors. Why? Because we cannot do anything with that of cell wall. Why? Because transpeptidase got altered and this D-alanine molecule get altered. We cannot do anything with that of two enzymes. The only other option is cell membrane in case of vancomycin resistant Staph aureus. So what are the drugs which comes under cell membrane inhibitors are like daptomycin, and polymyxins and daptomycin. So this is the counter action this is a counter action of doctors on the bacteria. So in this way we can inhibit the cell membrane synthesis, and this is a drug of choice for VRSA. So drug of choice for MRSA is vancomycin. So drug of choice for VRSA is cell membrane inhibitors like daptomycin. So there are some common drugs for the treatment of MRSA and VRSA like linezolid, like another drugs like linezolid and streptogramins, and the last one is tigecycline. So these are the another drug of choice for MRSA and VRSA. So this is about some resistance and the treatment of penicillins.